Good morning, class. Today, I will be talking about the chemical luciferin. So, why luciferin? And what is it? And where is it found? So, luciferin is a chemical behind bioluminescence. It is found in bioluminescent organisms such as some jellyfish, fireflies, and plankton. The name luciferin actually comes from the Latin word lucifer, meaning light bearer. So, each of these organisms that have luciferin actually have different variants of it. Their chemical structure and reactions involved are slightly different, but the main idea and concept behind them are the same. For example, existing varieties include firefly luciferin, ladia luciferin, and bacterial luciferin. As you can see, most of these types of luciferin are named after the organism that they are produced by. However, a few are given common names, and these include choleintrazine, vargulin, and foxfire, which are the luciferin types that are produced by squid, shrimp, and fungi, respectively. Until I researched it specifically, I actually did not know what exactly it was that caused this specific chemical to produce light. And really, who hasn't wondered about how fireflies produce their light as a child? So with that being said, we can move on to talk about the physical and chemical properties, reactions, and interesting facts about luciferin. So as I said before, luciferin comes in many different types. But however, the most common one is probably firefly luciferin. So that is the one that's going to be focused on today. So here, you can see the chemical structure of luciferin. It contains 11 carbons, 8 hydrogens, 2 nitrogens, 3 oxygens, and 2 sulfurs. Its IUPAC name is 4s2,6-hydroxy-1,3-benzothiol-2-ol 4,5-dihydrothiazole 4-carboxylic acid. So with that out of the way, we can look at some physical and chemical properties of luciferin. So of course, the standout property of luciferin is its fluorescence. So luciferin oxidizes or reacts with oxygen in the form of luciferase, an enzyme to form oxyluciferin and light energy. This will be focused on more in the reactions section. Things like the byproducts and catalysts again vary with each type of luciferin, but the concept is the same. The wavelength of light also varies with each type of luciferin, producing the different colors of lights that are seen from different organisms. For example, firefly luciferin absorbs ultraviolet light at a peak of 327 nanometers and emits regular light at a peak of 520 nanometers. So on the right here, you can see the image of different types of luciferin corresponding to the different colors of wavelengths of light that they produce. So the colors of light can also be slightly changed by some other external factors, such as the pH level. So now we can look at some general properties of luciferin. Its chemical formula again is C11H8N2O3S2. Its molar mass is 280.32 grams per mole. Its density is 1.8 grams per centimeter cubed. Its boiling point is 473.8 degrees Celsius at atmospheric pressure, has a flash point of 240.3 degrees Celsius, an index of refraction of 1.865, and finally an enthalpy of vaporization of 80.7 kilojoules per mole. So with the physical and chemical properties out of the way, we can now look at some reactions. So the two main reactions that will be covered here are the synthesis of luciferin and the reactions it undergoes to produce light. So first off, we'll start with the synthesis. So actually, outside of the last step, we actually do not exactly know how luciferin is synthesized in vivo or in a living organism. So this last step that we do know happens when the luciferin molecule is produced in a condensation reaction between D-cysteine and 2-cyano-6-hydroxybenzothiol. So this is actually the same step that we use when we produce luciferin artificially in labs. So now we can look at how the luciferin actually produces light. So there are actually two steps that are involved in producing the light. So the first step happens when the luciferin oxidizes to produce a peroxy compound called 1,2-dioxetane. After the 1,2-dioxetane is produced, the second step occurs. Since the 1,2-dioxetane is actually unstable, it decays spontaneously to produce carbon dioxide and an excited ketone. We actually call it an excited ketone because it is actually in a higher energy level than it would normally be. So, in order to get back to a lower energy level or its usual energy level, it emits light. So the final step in that reaction, the production of the excited ketone and its relaxation, is shown in the diagram above. So now we can move on to looking at some interesting facts about luciferin. So first off, we can start with its history. Bioluminescence has been noticed by mankind since ancient times. The first people who for sure documented it include Darwin and Robert Boyle. However, even people as far back as Aristotle seem to have mentioned it. But the aforementioned three were nowhere close to figuring out the source behind the bioluminescence, as the science of their day had not evolved enough. It took to the 19th century where a French pharmacologist, Raphael Dubois, had a breakthrough, and he demonstrated that the oxidization of a newly found chemical, which he named luciferin, was responsible for the bioluminescence. It then took another 10 years for a Japanese chemist by the name of Osamu Shimomura to isolate crystalline luciferin and finally produce its chemical structure in the crystalline Cyprindia luciferin, which was the name of his 1957 paper, for which he was awarded the Nobel Prize in 2008. So one other thing that I found extremely interesting was the efficiency of this reaction. So this reaction actually releases over 98% of its energy in light, which is in a completely different ballpark to our light sources today. The best light sources of today operate at a mere 2% efficiency. However, practical limitations stop us from using luciferin as a light source. For example, luciferin is expensive to make, and the fact that it needs to burn in oxygen to produce light make it a safety hazard and therefore cannot be used as an everyday source of light. Finally, luciferin has many applications, especially in biological research. It is used in genetic engineering to get other organisms like mice and worms to produce the necessary gene for bioluminescence. This you can see in the pictures on the right. This means that animals that generally would not be bioluminescent can be genetically engineered to do so. Luciferin is also used in imaging. For example, if a certain cell population is to be studied, those specific cells can be injected with luciferin to make them incredibly easy to find an image. So that more or less concludes my presentation on luciferin. Here are my works cited, and thank you for listening.